We are recording, just so you know. And also for anybody watching the video, this is going to be business as usual. You're going to get a series of attendance words. You're going to write them down as I say them. And at the end, you're going to send an email with the attendance words and with a screenshot of every time I send one of the attendance words. The first word for attendance is going to be ice, frozen water, ice. Just for the people in the video. That's your first word for attendance. And it is 7:38, so you can skip ahead about 20 minutes, and that's when the lecture will start. Listen, you get a little extra credit for reading the first one here. Morning.
Morning.
Since you're the second person here, third including myself, and you also get a little bit of extra pay.
Sweet.
Excuse me.
Dang. Voila. The whole class here.
Good morning.
So we just don't have like a lab to fill out this next week.
No pre-lab due today, right? We're still gonna meet for that next week, but we're going to have pre-lab due.
I don't do that. Don't fill it. How does it work?
You gotta pay attention. Just kidding, well, sort of. I did mention this last time, but actually I'll wait before we get started. I'll answer your question here in a second, but I'm gonna say it again, so I don't have to repeat myself. Wait till everyone else shows up. Or whoever else shows up. Obviously not everyone else is coming, I don't think.
My class is in turn. That's student's turn.
We've got less than 2 minutes, so a reminder for the two of you who are streaming right now, you will still need to send the words in before 9 a.m. But you do not have to send the screenshots. So every time I mention a screenshot, that's for the people watching the recording at a later date. So for those of you who are streaming right now, Michael and Kelly, you don't have to send the screenshots, you just have to send the words. But you do have to send the words, unlike everybody who's present. For you guys, it's not extra credit, that's proof that you paid attention. Let's see. 7:59, I guess I'll start attendance. Roosevelt is here, Champion is here. Gorby is not here yet. Gross is online. Gunther is not here yet. Randall's not here yet. Hill is here. Jackson is not here yet. Keys is not here yet. Back up. Gunther is here now. Anyway, Keys is not here yet. Reyes is not here yet. Sigmund, not here yet. Slaughter is not here yet. Smith is here. Stover is here. Tompkins is not here yet. Ines is not here yet. Excuse me, Watson is online. Young is here. Zweibeck is here. Lawrence is not here. Wilson is here. Did I miss anybody? It's 8 o'clock, so no one's late yet, not until you one. So let's do the normal reminders. And hopefully nobody's missing out on them, because it's the same thing I say every day. Attendance is mandatory and graded. So if you know anybody who's in this class and they don't show up, maybe you should remind them of that. Speaking of attendance, since today is National Talk Like a Pirate Day, first word for attendance is pirate. Of course, again, if you're here in person, you do not have to send those words, but you can for extra credit. If you're watching this live stream, you do have to send the words because that's proof that you're paying attention, not just logged on and taking a nap, which is what I would be tempted to do if I was logged in. And of course, if you're watching the video, you need to take this picture, the screenshot, as well as write down the word pirate. Which you guys probably won't get this joke, but what is a pirate's favorite amino acid? Arginine. So I guess you have to know all the amino acids to know what I mean with that one. But anyway, jokes aside, it's 8:01. Let's get started. Another announcement that I guess I should say this every day too, because you should really check your WVS emails and your course announcements every day. Very important information is being passed along there. This is mostly for the other courses. You guys, I get to see you every Monday, Wednesday, Friday, but my online courses, people sending me the dumbest questions because they'll say the same thing over and over in the announcements and in the emails. I send an email asking about it. Sigma, right?
Yes.
Okay. So anyway, yeah, make sure you're checking those daily. There is no pre-lab due today. As I mentioned on Wednesday, next week's lab, or the next lab we do is going to be the photosynthesis and respiration lab. It's a very important one because you also have to write a lab report about it. So since it's so important, next week for lab, all we're going to do is meet and have the pre-lab lecture. So I'm going to explain this lab in thorough detail next week. I'm also going to explain how to write a lab report next week. That way, the week prior to that, we're just going to come in and hit the ground running when we do the photosynthesis and respiration lab. So again, there is no pre-lab due today, but we still have lab. Also, like I said at the beginning of the semester, you have to go ahead and plan on being here every Wednesday, right? So I said at the beginning of the semester, that first Wednesday of the semester, and then of course the last Wednesday of the semester, those are the only times you should plan on not being here. Otherwise, you should plan to be here. So any questions about the pre-lab situation or next week's lab? All right, and again, since all we're doing is a pre-lab lecture next week, that means you also don't have anything to do. Like all you have to do is show up and pay attention. So that's hopefully it gives you a good week off to, relax and get yourself prepared for a more important lab that we're going to do in 2 weeks. But make sure you're here on time, because since we're not doing anything next week, you have nothing to turn in next week, your attendance is based on being here on time and not leaving early. So, make sure you're here. So any questions about pre-labs? All right, to remind you, Lab 4 is past due today. You have to turn it in via WVSU Online. Most of you have got that, some of you don't. If you don't know how to do that, meet with me so I can show you, because you're going to get zeros until you do that. So let me know if you need help. Also, as you hopefully, if you read your emails and announcements, you saw my post about the mistakes. It kind of sucks that there was those mistakes, but I'm kind of happy about it. One of the things I love about these mistakes, especially in labs, is it's teaching you. This is a really good thing, because most of you probably aren't going to be doing biological experiments after you leave this class. So in that sense, doing biological experiments is very unimportant. But what you will be doing, hopefully, as an educated citizen, is voting and sitting on a jury, maybe even running for office yourself, right? You're going to be a leader, or at least making decisions on who's going to lead. And hopefully because of that, or hopefully because of this, you'll be better at doing that, right? So again, just reiterating what I said in that announcement, someone should have seen those red flags, right? The fact that data said there's no water in the beaker, someone should have been like, that doesn't seem right. Let me double check that. And this is exactly what you should do once you leave this class and once you've graduated and someone says, I can't believe you're voting for so-and-so. So-and-so supports this. Let me show you this study on this. And then look at the study and look for red flags. Like, that doesn't look right. Something's wrong with this study. There's something wrong with this data, right? So that's the kind of thing you should be looking for. So it's a good learning moment. No one lost points for the fact that the dye supposedly shrunk, which is against the laws of physics. No one lost points for that. But you did lose a point that you can fix to get your point back for saying things like, the water was not in the beaker at the end, things like that. So again, these mistakes are good in a sense because they're a good learning moment. Hopefully that'll help you remember things. And that's the things you need to be looking for, right? Look for red flags when you're looking at data. Anyway, any questions about that? Again, it's past due today, right? So you have to get it done today. If you need help, let me know. I'm on office hours until noon today. I can help you do it. And what else? Oh yeah, the other thing with the table. I don't know if I mentioned this in the announcements. Just do basic math, right? If something started at 20 and ended at 10, that's a change of negative 10, right? Or what's another example? If something started, and the other one's a little bit more complicated with the table 3, right? Just do basic math. If you want to know how much something changed, figure out what its last number is and subtract the original number. Like if it was weight, like if I weigh 250 pounds now, which I probably do, unfortunately, and I weighed 225 pounds a year ago, and I wanted to know how my weight changed, I would take 250, I would subtract 225, and I would be left with 25. I'm like, oh, I gained 25 pounds. My weight changed positive 25. Hopefully that's basic math, right? You take the last number, you subtract the first number, and that is your total change. But nonetheless, if you need help with that and forget, just brain dump what I just said. I'm here to help you. Meet me online. I can help you do this. So any questions about that for? All right. Again, just as a reminder, exam 1, you can retake it, even those people who never took it in the first place. And remember, don't forget, you have to tell me once you're done retaking it so I can go in there and manually change your grade. Otherwise, I don't know you retook it and your grade won't change. So let me know if you've retaken the exam. And then finally, again, to remind you, I'm here to help. Does anybody have any questions or any of your own announcements or reminders? All right, let's jump back into it. We were talking about enzymes. Enzymes are a very important concept, not just for this exam, but because a lot of the stuff we're going to talk about for chapters upon chapters upon chapters deal with enzymes. Up until the end, right? The last portion of the chapter, that's when we stop talking about cells, and that's when we start talking about like a bunch of organisms, right? We start looking at the big picture at the end. But until then, we're mostly looking at cells. And if we're looking at cells, no matter what we're talking about them doing, it involves enzymes. Last thing we talked about with enzymes was this, activation energy. And of course, that's the graph from your textbook, which I don't like, because it's a little bit too technical. This is a better representation, in my opinion, that we've already seen. Right? This is from the Yoda textbook. This just shows, this is the same chemical reaction on the left and right. It's the same thing. You start with the same reactants, you end with the same product, you're releasing the same amount of energy, right? The starting energy is the same amount as the ending energy. And that's what this is all about, right? Either putting off energy or taking energy. The idea of an enzyme is it reduces that amount of energy that you need to invest to get that reaction started, which is important because in our cells, a lot of times this activation is so high that the chemical reactions that are happening that are keeping us alive just wouldn't happen at all, right? So without these enzymes, we just wouldn't be alive because there wouldn't be enough energy for us to have these chemical reactions. So that's the last thing we talked about. And that's probably the most important thing you need to know about enzymes. It's like, what is their purpose? Their purpose is to lower activation energy. Remember that before you memorize the rest of the stuff. The rest of the stuff I'm going to teach you is basically how they lower the activation energy. So here's some words you need to know. Well, first of all, before I give you those words, one thing you do need to know is that enzymes are very selective. Meaning, all these little chemical reactions that we're going to talk about throughout this semester, that each different chemical reaction has its own enzyme, generally speaking. Generally speaking, there's one enzyme for each chemical reactions. And there's thousands of different chemical reactions happening. So these enzymes, again, they're very selective, very specific. Even though it's not written down on the screen right now, I mentioned it earlier and I'll mention it again. The way they are so selective is it's all about the shape, right? It's protein enzymes, as far as we're concerned, are proteins. And all proteins, it's all about the shape, especially enzymes. It's very much about the shape. So with that being said, I'm going to introduce you to some new words that you need to know for the exam. First of all, the word substrate. So enzymes recognize the substrate. So again, remember an enzyme basically makes the chemical reaction happen. So in that context, the substrate is the reactant, right, or reactants. In this picture, we're taking one molecule and breaking it into two. So in this case, in this picture, the substrate is just one molecule. Sometimes it can be more than one, sometimes it's just one. The point is, The enzyme acts on the substrate, right? The substrate is what the enzyme is working on. It's the product. Now, there's a specific site on the enzyme when this happens. So it's not like you just touch the enzyme in any old place. It has to happen in a specific spot. And that is called the active site. And you're going to see a lot of different pictures in this chapter. And they're all going to look slightly different, but they're all basically going to look like this. It's going to look like a blob with a certain shape, with some other thing that fits that certain shape going into it, and a chemical reaction happening, right? So again, here, this is a good example of actually see if anybody remembers for extra credit. Would this be a catabolic or anabolic reaction that's on that picture right there? No one wants to guess for extra credit? The one at the bottom, what kind of reaction are we looking at? Catabolic or anabolic? Catabolic, good. You said it? All right. If you send your words in today, remind me that you said that. Yes, it's catabolic. Remember, because catabolic breaks things down, anabolic builds things up. One easy way to remember that is if you ever heard the term anabolic steroids, even though they're used for legitimate reasons, a lot of times they're used for people trying to build themselves up, right? Anabolic builds up, catabolic breaks down. And so someone sent me an email, and it reminded me of something I used to think of when I was a student, because I don't really like cats. Sorry if you do. But whether you like them or not, you have to admit they like to break stuff. They get on your counter and just look at the base and look at you, and like, boom, hit it over, right? So when I think of breaking stuff, I also think of cats. Not because I want to break cats, but because cats break stuff. But anyway, if you think of breaking stuff, you can think of cats, catabolic. If you think of the anabolic steroids building up, that's anabolic. So anyway, yes, the active site is where all the business happens, right? The active site is where the substrate fits into the enzyme. And that fit has a word. It's called the induced fit. Now, I'm probably not going to test you on any of these terms, but I'm going to use these terms in a question, right? So I'll be asking you questions about, hey, if this thing's happening, what are the results? And to be able to understand that, you need to know what a substrate is in an active site. Now, induced fit. An induced fit is the entry of a substrate, when the substrate enters the enzyme changes shape, and then it kind of makes it more snug. Excuse me. And I use this picture as an example because there we've got people hugging, right? And in a sense, it's kind of the same thing, right? You change shape when you hug because before you're embraced in the hug, you've got your arms open, right? You just like a regular old enzyme, waiting to do your thing, waiting to make some chemistry happen. Then you do the hug, someone comes in chest to chest, and you guys wrap your arms around each other. That's sort of like what an enzyme does, even though you can't really see it very well in that picture. It has more of an open shape. Like, come on, let me get my substrate. And then once the substrate comes in, it kind of, I don't want to say squeeze, but you could say that, it kind of squeezes around it. It's an induced fit. So yeah, make sure you're very familiar with these words. Oh yeah, one thing I didn't mention about the active site. Let me back up, sorry. So again, as you can see from these pictures, the substrate fits the active site, right? It's very much a thing of the same shape. And like I was saying earlier, and I somehow got sidetracked, you're gonna see a lot of these different pictures. They're gonna look different, but they're gonna be slightly the same. One of the things they're gonna have in common is, again, you have a blob with a specific shape and a molecule that happens to fit that shape. So that shows, and it's accurate, that enzymes are all about the shape. But another thing, and we're not gonna talk about this much, but it's also a little bit about the chemistry. So for example, if there was something about this enzyme that gave it a positive charge right here in that part of the enzyme, then you could expect this probably has a negative charge right there. That would mean it's a good chemistry fit, right? Because negatives and positives come together. Or another example is if this part right here of the enzyme is polar, right? Like hydrophilic, right? If that's polar, it's hydrophilic, then you probably guess this thing is also polar and hydrophilic, right? Because hydrophilic things come together. Hydrophobic and hydrophilic things repel each other. So again, it's not just the shape, but it's also the chemistry. The chemistry is good. Any questions about the slide? Okay. So again, the substrate comes into the enzyme. You get the induced fit. The enzyme does its job, whether that's breaking down a molecule, like you get a catabolic reaction, or building a molecule, as in an anabolic reaction, whatever the case may be. The substrate comes in, the juice fit happens, it does its job, and then it releases the product, right? So then it's just free to do its job again, right? It's got its arms wide open again, so to speak. It's then able to accept another substrate. And this is very important because that means it's not used up in the reaction. And the reason I say this is very important is I'm teaching to the exam, unfortunately. So in the exam, I'm going to give you a reaction that you probably never heard of because I'm probably just going to make words up. It won't even be a real chemical reaction. And it's going to say, this plus this gives you this plus this, or whatever it may be. Now, I'm going to give you that chemical equation, then I'm going to say which one of these things is the enzyme. And even though it's going to be a bunch of made-up words that you've never heard before, if the thing is on both sides of the reaction, which I could draw it, maybe just x plus y plus z, imagine those are molecules, not letters, gives us, I don't know, this is really hard to draw with, x plus a. If you saw that equation on the exam and I said, which one of those is the enzyme? The answer is X, even though it's really hard to read. Because X is on both sides of the equation, right? Enzymes are not used up in the chemical reaction. Actually, a lot of times they're not even put into the chemical equation. But when they are, you're going to see them on both sides of the equation because they're not used up in the reaction. So make sure you know that. This next bullet point is important, and we've mentioned it before, which we weren't specifically talking about enzymes. When we were talking about proteins, I mentioned that proteins are sensitive to their environment. We talked about why, right? We talked about temperature, right? As you increase the temperature, that means what you really do is increasing molecular movement, which means it's easier to break those bonds that are holding it together, especially hydrogen bonds. Those are the weakest of the bonds that are easier to break. So if you heat things up, it's more likely to melt. Or the proper term for this in biology is called denaturing. When a protein melts, it's denatured when those loose bonds are broken. So yeah, temperature is one of them. There's an optimum range. I'm not going to test you on that. But enzymes are designed to work in an optimal range, which makes sense. So the enzymes that we have in our body have an optimal range, and they're probably very different, for example, than the enzymes of the bacteria that live in the hot springs in Yellowstone National Park. Or not the hot springs, but the geysers, right? Those things are living in boiling water. So you can imagine their optimum range is a lot higher than ours. You can imagine if we put our enzymes in that environment, they would probably denature, right? That's what happens when it's too hot. Also, another thing that changes it is the pH. We mentioned that earlier, again in discussion, in chapter 2, when I showed you a picture of this enzyme or this protein. We had zoomed in and you saw these hydrogen bonds between this partially positive hydrogen and this partially negative oxygen. Anyway, that's a partial bond, right? Those are partially charged. They have that weak hydrogen bond in it. With the pH, remember, if it's too acidic, that means you're putting in a bunch of H pluses. Those are fully charged ions. If it's too basic, you're putting in OH minuses. Those are fully charged ions. So as you can imagine, those things are going to disrupt those hydrogen bonds and again cause it to basically melt. Another thing that affects them is salt concentration. That's kind of the same thing as far as we're concerned because again, we're talking about ions, right? Positively charged things, negatively charged things, that's going to disrupt hydrogen bonds. And also, they're sensitive to coenzymes. And for now, you don't need to worry about coenzymes. I'm going to describe them later. And even when I do, I don't think I'm going to test you on coenzymes. All right, one more thing with this bullet point here, this little equation. This is going to help you. Another thing about this fake chemical equation you're going to get on the exam is it's not going to be x plus y plus z, right? There's going to be words. So with those words, the one that ends with ACE is the enzyme. So there's going to be a bunch of words you didn't understand, but the one's going to end with ACE, and that's going to be an enzyme. For example, one of the enzymes you're about to learn about is something called ATP synthase. It ends with ACE. It's an enzyme that makes ATP. You'll learn about it later. So if it ends with ACE, actually the next one you're going to learn about is called lactase. It's the enzyme that breaks down lactose. So again, in this exam, when you get some chemical reaction that you've never seen, and I ask you, what is the enzyme? First of all, it's going to be on both sides. Second of all, it's going to end with the word "ase," or it's going to end with the suffix "ase." Everybody understand that before I teach you one more thing about this? All right, they're also named for their substrate. So using a real life example, this wasn't X plus Y. If it said, let me see, lactose plus lactase equals glucose plus lactose, galactose plus lactase. Anyway, I guess it'd be easier if I wrote it and you can see it. The point is, they're named after the substrate, right? So in this example I just gave, hopefully you can figure out lactase is the enzyme because it ends with ase and it's on both sides of the equation. But then you just look at the rest of the chemical equation, like what word looks like the enzyme? Lactose looks like lactase, therefore lactose is the substrate. So again, you will get an exam question where I give you a made-up equation, and I'm going to ask you which one is the enzyme, and I'm going to ask you which one is the substrate. And again, enzymes on both sides of the equation, it ends with ase. Enzymes are named after their substrate or what they do. And I'll give you some more examples as we go forward. So for now, that's all there is to say about that. Are there any questions about that side? All right, here's what I was talking about. This is a picture from the old textbook, not the one you guys are using, but it's a great example, and it's a real life example. This isn't just some made up thing that I'm going to use on the exam. This is an enzyme called lactase. As you can imagine by its name, it works on something called lactose. Here's its active site. It's a perfect fit, both chemically and shape-wise, for the disaccharide lactose, right? Lactose is made up of galactose and glucose. Not that you need to know that for the exam, it's just an example. But they're bonded together to make a disaccharide. It fits into this enzyme. This enzyme's job is to break it down, so a catabolic reaction, right? And there's something about that enzyme that makes breaking that bond easier. Maybe it changes shape. Maybe it changes the chemistry. Doesn't matter. And just the point is, it makes it easier to break that bond. It lowers the activation energy, right? We have some water coming into it because, again, it's a catabolic reaction. We're breaking something down. Also, like you learned a long time ago, way back in chapter 2, these catabolic reactions are also called hydrolysis reactions, right? When you're breaking something down, it's hydrolysis. When you're building it up, it's dehydration. So anyway, it's ready to go. It does. It breaks this lactose down into its two parts, which are glucose and galactose. They go their own way. Now that this enzyme is free and ready to do its thing again. So that's how it works in a nutshell. Any questions about that? All right, now to complicate things, it's not always that simple. There are these things called enzyme inhibitors. Does anybody know outside of biology what the term inhibition means, right? If someone says, oh man, I went to a party, I drank a little bit too much, and I lost my inhibitions. What does that mean, inhibitions? In that context, when someone says they lost their inhibitions, what do they mean? Keys? What does that mean? Lost their inhibitions. I know somebody knows. You guys are just too shy. All right, well, I guess I'll just say it then. Enzyme or inhibitions, right? So when someone says that in that context, oh, I lost my inhibitions, that means they lost the thing inside of them from keeping them from doing stupid crap, right? So, you know, when you're sober, you're usually like, hey, maybe I should take my pants off and jump off the roof into the pool. Your inhibitions kept you from doing that stupid thing. And you go into a party, you drank too much, you lost your inhibitions, you did the stupid thing. Point is, inhibitions are things that keep you from doing something, right? An inhibitor, no matter whether we're talking about biology or not, it's something that stops you from doing something, right? It prevents you from doing something. So an enzyme inhibitor is something that inhibits metabolic reaction. In other words, it stops that chemical reaction from happening, or at least slows it down, or makes it harder for it to happen. Because these enzymes, again, they're catalyzing these chemical reactions that are constantly happening in our cells because we're trying to make a product, right? We've got these reactants, we want to turn them into the products. However, we don't always need that, right? Your cells might not always need that, whatever the product is. So there needs to be a way to slow those enzymes down or sometimes stop them temporarily from doing what they do. And that's what inhibitors are for. So you need to know what an inhibitor is, and you need to know basically how they work. And basically how they work is they bind to an enzyme. They literally attach themselves to the enzyme. And by attaching themselves to the enzyme, it disrupts the function, you know, whether temporarily, usually temporarily. So does anybody have any questions about this first main bullet point? You know what enzyme inhibitors are and how they work, right? They stop these reactions from happening, or at least slow them down. And they do it by attaching themselves to the enzyme, which disrupts the function. That's what they are. That's how they work. Now, to make it even more complicated, there's two subcategories of inhibitions of enzyme inhibitors. The first one is competitive inhibition. And this is when the inhibitor plugs up the active site. This is like in that picture right there that I did not make up, I just found it. But yeah, you can see that yellow thing is the enzyme, that blue thing is the substrate, that green thing is the inhibitor, right? It's not even the same exact shape as the substrate, but it's the exact same shape as that one part of the active site. So it plugs it up. That competitive inhibitor basically literally just physically blocks the active site from meeting up with the substrate. That's what a competitive inhibitor is. You need to know that. Competitive inhibitors plug up the active site. And a competitive inhibitor is one of the two types of enzyme inhibitors that I'm about to teach you. Any questions about that one before I teach you about the next one? All right, let's see if anybody can guess this. This one's obviously, it's going to be pretty easy, otherwise I wouldn't guess. See if you guys can guess it. So if one of them is called competitive inhibitor, any guesses what the next one's called? There you go. If you send in extra credit, if you send in your extra credit words, remind me that you guessed that. Yes, non-competitive inhibition. I wish all biology was like that, where all the words just made sense. It'd make things a lot easier for students. Anyway, non-competitive inhibition is when it binds at a site other than the active site. So it's binding, it's still binding to the enzyme. Because as I mentioned when I first mentioned inhibitors, that's how they work, right? They bind to the enzyme. Competitive inhibitors bind to the active site. Non-competitive inhibitors bind somewhere else. That's all you need to know. So how does that work, right? If you're not blocking the active site, how does it work? It works because it changes the shape. And that's all you need to know. That one bullet point is like a whole chapter in some classes, right? Like how exactly do these non-competitive inhibitors change the shape of an enzyme? And as you might imagine, like I mentioned earlier, enzymes are very selective. So there's thousands of different enzymes. Therefore, there's thousands of different inhibitors. They all work a little bit differently. But basically, this is how they work, right? They change the enzyme shape when it's a non-competitive inhibition. So it isn't that they blocked the active site, it's just they changed the shape. But remember, the shape has to fit. So does the chemistry. But yeah, if you change the shape, then it won't fit in it, and therefore you've inhibited the function. Another word for this is called allosteric inhibition. Let me check on that noise and make sure we're still good. I don't want it to stop recording again. I've been having that issue. Bear with me. I guess while I'm doing this, I'm going to give you the next word for attendance, words, if you will. POW/MIA. Because today is POW/MIA day. All right, we're still recording. I heard a noise. All right, well, anyway, if anybody's online and you got questions, I don't know, you can try to type in the chat box. I might hear it, but I might not. Anyway, let's get back to it. Go. All right. So, any questions about this before I give you an image? I'm going to show you a picture that basically shows you all this. All right. So here we go. Here's an enzyme that works normally, right? You've got the active site, the substrate, it fits, no problem. Here you have an enzyme inhibitor that's actively, right? So this is plugging up the active site. You can see this inhibitor is not the same exact shape as the substrate, but it's the right shape to block that active site, so it keeps it from doing its thing. And of course, you got the allosteric version, which is where the active site itself is not blocked, but once this inhibitor binds somewhere else, it changed the shape of the active site. So now the substrate can't fit in the active site. So there you go. It's pretty simple stuff. Competitive inhibition, non-competitive inhibition. They both work the same way in that something binds to the enzyme and it keeps it from doing its job, which is making a chemical reaction happen. Any questions about that picture? All right, let's make it even more complicated. Enzymes often require helper molecules, right? So a lot of times they're not just doing their own thing on their own. Sometimes they need help. And in a sense, what we're talking about now is kind of like the opposite of what we just talked about, right? We talked about inhibitors, which are things that are keeping enzymes from doing their job, or preventing it, or slowing it down, whatever the case may be. Now we're talking about the opposite. These are things that the enzymes actually need to help it work. There's two different types. I'm probably not going to ask you to know the difference between the two on the exam, but they are cofactors and coenzymes. Cofactors are basically ions, right? They're inorganic ions. So things like iron and magnesium, all those things we briefly talked about chapter 2 when we talked about those trace elements that you need, and those the smaller amounts of nutrients that you need. That's the cofactor. Same thing with coenzymes. These are organic molecules, so they're bigger. They have carbon and hydrogen based, which is why they're organic. Excuse me. Thanks. Woo. Anyway, whether it's a cofactor or a coenzyme, they both participate in reactions without being altered. So that means they're recycled and reused just like the enzymes, right? And that's actually very much the role of vitamins, right? So when people say, hey, you need vitamin C, vitamin D, whatever, right? All these vitamins they say you do your diet, very often that's how they're used. So that's something you could look up for extra credit if you want to write a paper about that. Like pick a vitamin, see if it's used as a cofactor or a coenzyme. It probably is. Of course, it might be hard to find, but you can look into it. Excuse me, any questions about this? Excuse me. All right, so going back to inhibitors, matter of fact, I should probably swap the order of these slides, because I guess we weren't fully done talking about inhibition yet. But there's something called feedback inhibition, and this is what I was hinting at earlier. This is the use of a product to regulate its own further production. Sorry, man, I thought I was over this cold, but I guess I'm not. Anyway, so remember like I was saying earlier, before I even told you what an enzyme inhibitor was, I kind of hinted at its purpose. Like the cells constantly doing these chemical reactions, constantly making these products, but they don't always need the products, right? Your cells are like factories building a bunch of different things. Are you old enough to remember fidget spinners? Okay. You remember those things were huge for a while. You guys, like everybody had them, everybody wanted them. That was a big thing, fidget spinners. Now what's those things that they're selling, those ugly little things that are like going for thousands, those little furry things? What are those things called? Boo-boo things or Lululemons? I don't know. What is it, Labubu? Okay, there you go. So imagine you're a toy factory, right? And back in the day, there was fidget spinners. You're like, all right, everybody stop what you're doing, right? So you take most of your material, most of your employees, and you stop making other stuff, and we're going to make a bunch of fidget spinners because it's making us a ton of money. This is what we need right now. And you did. And then that fad went away, so you stopped making them and had to make other things. And now you're doing the boo-boo or whatever you just said, LaGrange, I don't know. So now if you're running this factory, you're like, all right, we're going to make some of these LaBoo-boos. If I'm saying that correctly, right? The point is, if you're a factory, you don't always need to make the same stuff, the same amount of stuff, right? Sometimes you got to have more of this, less of this. Same thing with your cells, right? Your cells need to make products, but they don't always need to make it. And that's what feedback inhibition is. So you have these chemical reactions happening, you keep producing this product, and eventually, if you have too much of that product, it's going to start interacting with those enzymes again, and it's going to prevent the enzyme from making more of it. So in other words, think back to what you just learned about inhibition, right? So you have this enzyme inhibitors that stop enzymes from doing a job. Well, in this specific case, it's the product of that reaction that is also the inhibitor, which makes sense because again, if you're producing this certain molecule because the cell needs it, it's going to use it up, right? So you don't run the risk of that molecule inhibiting your enzyme. But if you have enough of that molecule, and your cell is not going to use it up, and it's going to be in excess, and because of that, because there's so much of it floating around in the cell, it is going to come back, and it is going to meet the enzyme, and it's going to inhibit it from making even more of it. So, any questions about feedback inhibition? It's just inhibition, like I taught you earlier, except now it's the specific case of the product is the inhibitor. Then, of course, feedback inhibition could be competitive, it could be non-competitive. In this picture, let me see. It's the substrate, enzyme 1, first intermediate substrate. We haven't even talked about intermediate substrates. I don't like this picture. So yeah, it turns out, not that you need to know this, but like I said earlier, enzymes work on substrates, but sometimes there's multiple enzymes working together. Sometimes there's multiple substrates working together, but we don't worry about that in this 100 level course. But anyway, that end product, yep, just as an example, this is a non-competitive inhibitor, right? Because it's not blocking the active site, it's binding somewhere else, which changed the shape, which kept it from doing its thing. Anyway, that's actually it for enzymes and enzyme inhibitors. Is everybody good on enzymes before we move forward? It's a very, very important topic. One, you're going to get a bunch of questions about it on the exam. Second, again, for most of the semester until the end, we're going to be learning how cells work. When we learn about how cells work, almost everything that's happening inside of cells is happening because of enzymes. Very important concept. All right, now I'll move on to the next thing. Next, we're actually going to talk about respiration. So I will tell you this, like I said before, this exam is almost entirely about energy. Like if we're talking about the properties of life, this exam 2, that we're going to have in about 3 weeks, it is almost entirely about energy, right? This transformation of energy from one form to another. That is one of the properties of life. This is what the exam is about. Almost the entire exam, other than a few things that we've talked about previously, like potential energy versus kinetic energy and 34% of your energy being put to work, whatever, all that stuff. Most of it is going to be about photosynthesis and respiration. And in most of these questions, or most of the questions on the exam about photosynthesis and respiration, I'm going to ask you to tell me when this happened. So I'm about to teach it to you, but just back up and look at the big picture. Respiration, what you're about to learn, is broken down into 3 phases. There's glycolysis, there's citric acid cycle, and there's oxidative phosphorylation, which we also call the electron transport chain. Those are the 3 stages. So a majority of the questions about this respiration, which is going to be about half of the exam, is going to be saying this. In which of these stages did this happen? In which of these stages did that happen? In which of these stages was this used? In which of these stages was that produced? So as we go through this, keep in mind, that's how the test questions are mostly going to be. It's going to be me telling you this thing and you telling me which of those 3 stages it happens in. So let's talk about it. The learning objectives in this section are, at the end, you'll be able to explain how ATP is used by the cell as an energy source. Matter of fact, in my opinion, if you're studying, that's the first thing you need to learn for this section. You need to know, like, what is the cell's main source of energy? ATP. Memorize that before you memorize anything else. Because everything else I'm about to teach you is how the cells make ATP. So if you can't remember that the cells need ATP for energy, but that's the main source of energy, then the rest of it is even that. Speaking of which, don't get confused. There will also be a test question that says, what is the ultimate source of energy on Earth? And the answer is the sun. That's the ultimate source. The specific source, right? What's the specific thing that cells use for energy? It's ATP. Anyway, by the end of this section, you will also be able to describe the overall results in terms of molecules produced at the breakdown of glucose by glycolysis. So again, I'm about to teach you respiration, which is broken down into 3 stages. Glycolysis is the first stage. So you're going to be able to tell me what happens in glycolysis, right? Also, I'm also going to do a little backup eventually, where we're going to look at the big picture, and then we're going to zoom back in and look at these specific, smaller pictures. Anyway, let's talk about ATP then. So again, no matter what the chemical reaction is, whether it's giving off energy or taking in energy, whether it's exergonic or endergonic, regardless, it needs that input of energy, right? It needs that activation energy. Now, granted, the enzymes are lowering the activation energy, like we just talked about, but you still need some. And where does that energy come from? It comes from ATP. Adenosine triphosphate. You do not need to know what ATP stands for. So there would be no test question where I say, what does ATP stand for? But you do need to know it just in case, for the exam, I might use the full word adenosine triphosphate in a question, or I might use ATP. I guess the point is you need to know it because you need to recognize it when you read it. But again, there'll be no question where I ask you what ATP stands for. This is a very small, simple molecule. It is the molecule that contains the potential energy for cellular work. Remember when I first taught you about potential energy, I also mentioned other types of energy, one of which was chemical energy, right? Chemical energy is a type of potential energy. It's however much energy it takes to build a molecule. That's how much energy is released when you break it. Hence the potential energy in ATP. It is the primary energy currency in cells. So again, that would be a test question. Like, what is the primary source of energy in cells? It is ATP. And for extra credit, you can look up what are the other sources of energy. There are a few other sources, but ATP is the main one. It powers a majority of the energy requiring cellular reactions. As far as we're concerned in this course, it basically powers all of them. But again, for extra credit, you can look up what are the other ones. What are the minority of energy requiring cellular reactions that don't use ATP? Actually, you're going to learn about one of them in this chapter, but anyway. And I like this last bullet point. I don't remember if I wrote this or if it's from the textbook, but it's a great way to look at it. They're basically like rechargeable batteries for your cells. Your cells need energy, and they get it from ATP, and ATP is basically like just a rechargeable battery. And even though I'm going to talk about this in detail later, I'll just go ahead and tell you now. In your cells, the mitochondria is basically the battery recharger, right? That's the thing that's recharging your batteries. So like I said previously, you can think of your cells as factories. They're building a bunch of things. All those machines in the factory, they need energy, right? They run off of batteries, so to speak, and the batteries are in your cells. That's ATP. And when they need to get recharged, they go to the mitochondria, where they get recharged, and then they go back into the cell, do their job until they get dead again. They go back to the mitochondria, so on and so forth. Any questions about this side? All right. I'm probably not going to get this detailed. No, I'm definitely not going to get this detailed on the exam. But I do want to talk about its structure because, like I mentioned a lot of times this semester, there's a big relationship between structure and function. So let's talk about this. This is an adenosine molecule. Hopefully you recognize that word, but you don't necessarily need to yet. But remember when I introduced you to nucleotides, I taught you about 4 different nucleotides, A's, C's, T's, and G's. Well, the A, that's this. So the nucleotide adenosine is also used to make ATP. And I'm going to test you on that, but I'm hoping you recognize that word and say, hey, that sounds familiar. And it is familiar because it's the same thing. So it's a nucleotide, because remember, a nucleotide had a sugar, a base, and a phosphate group. But it only had one phosphate group, right? ATP has 3 phosphate groups. That's the charged battery. ATP is your charged battery. Now, when it's used, you lose one of these phosphate groups, and then it's called ADP. So it's not triphosphate anymore, it's diphosphate. The only reason I'm bringing this up, again, I want to talk about structure and function. Because remember, I said however much energy it takes to build a molecule is how much is there to be released when you break it. Well, it takes a lot of energy to build this thing. Let's look at this. Remember, first of all, I told you on the outside of atoms, we have electrons, right? Electrons repel each other. So that already makes it hard enough to bring atoms together. Then we have this. These things are negatively charged, right? Negatively charged oxygens, all those negatively charged oxygens. So they're really repelling each other. You try to squish those together, it's going to take even more energy. Then, and I haven't taught you this, you don't necessarily need to know it, oxygen itself is a very electronegative atom. So right there, you have more reason they don't want to be together. Then you've got these double bonds. Double bonds are also very electronegative, right? Because that's 2 pairs of electrons. So everything about these phosphate groups, they don't want to be together. They're negative, they're very negative, they repel each other. So it takes a lot of energy to squish them together. Therefore, when you take off one of those phosphate groups, you're going to release a lot of energy. So any questions about the relationship between ATP, which is the charged battery, and ADP, which is the dead battery, so to speak? Okay. Here's a picture from the old textbook that shows basically how it's used. So here again, you have ATP, right? You have those 3 phosphate groups. You knock one of them off to release some energy, and now what you're left with is ADP. and that phosphate went off and did its own thing. So again, fully charged battery on the left, you use it up, and now you're left with a dead battery on the right. Triphosphate is the full battery, diphosphate is the extended battery. Of course, you could look this up for extra credit too, and this one be even, you'd probably have to dig pretty deep. What about adenosine monophosphate? Which hopefully, as you just remember, even though I didn't say it in those terms, that is basically just the A, the nucleotide A. But also, yeah, what happens if there's just one phosphate? Also, is there any such thing as an adenosine with 4 phosphate groups? Does that ever even happen? It's things you can look into for extra credit if you want. Anyway, again, we're talking about how ATP works. ATP works by transferring that phosphate group. Now, for the exam, though, I'm not going to get that technical. You can just remember that ATP works by giving up one of those phosphate groups, right? When you break those chemical bonds, it releases that energy. That's all I care for you to remember. But yes, technically, that phosphate doesn't just go flying around and go wherever. It actually goes somewhere. It is actually transferred to somewhere else. But again, for the exam, just remember that you break that bond, you lose one of those 3 phosphate groups, and that releases energy. And I give you some pictures to show what I'm talking about. These are pictures from the old exam. Here's ATP used to make your muscles move, or ATP being used to make your muscles move, right? So obviously when you move, that uses ATP. So this ATP, you transfer a phosphate group to this motor protein that causes it to move, and then of course you're left with ADP and a phosphate group. There we go. This is something we've talked about earlier. We talked about active transport, right? I said active transport is when things go from an area of low concentration to high concentration, which is against the fusion, right? So it requires an input of energy. Like I said, when we talked about that, where does the energy come from? It comes from ATP. So it transfers that phosphate group, that releases energy, and then now you have the energy to pump these purple balls up their concentration gradient. Again, that's just an example. And then one more example, and this is one we talk about more than anything else, is this idea of building molecules or sometimes breaking them down, right? So we want to put this X and a Y together, right? It takes energy to put molecules together. Where does that energy come from? It comes from ATP. ATP gives up that phosphate group, transfers the phosphate group. Now we have the energy to squeeze that X and Y together, and now that's our product. And now we're left with the dead battery and a phosphate. Any questions about that? All right. Your cells are spinning ATP continuously. It's basically what keeps you alive. And they are recycled when the ADP and the phosphate combine. So basically, again, like I was saying earlier, ATP is the charged battery. ADP is the dead battery. And the whole purpose of cellular respiration, which is what I'm about to teach you next week, this would be a good place to finish this up. But anyway, this is what it's all about. It's all about recharging that battery. It's all about turning that ADP back into ATP. Everything you're about to learn, that's the purpose of it. And here's a number I might ask you. About 10 million ATP are consumed and recycled every second in one single working muscle cell. So think about that. 10 million ATP in one cell of your body per second. So think about how much ATP you go through. That's just one cell. And you can look that up for if you want some other numbers, like how much then does your whole body use in 1 second? About a whole day. How many calories is that, right? How many calories does it take to make 10 million ATP? But anyway, let me finish this up. Again, ATP is the charged battery, ADP is the depleted battery, and your textbook talks about this thing called the ATP cycle, which is basically just the cycle of, oh, I've got this charged battery, I'm going to use it till it's dead. Now I have a dead battery, I'm going to recharge it till it's full. Okay, now I have a charged battery, it's that simple. And you're going to learn about a few cycles in the next 2 chapters. This is the easiest one, because that's really it. You got your charged battery, your dead battery, charged battery, dead battery, that's the cycle. Here's a picture that's not from your textbook, right? So you got ATP, that's your charged battery, you use it, it releases its energy that gets used for work. Now you're left with a dead battery. The whole purpose of cellular respiration, which is what you're going to learn about next week, is to bring these things back together, right? The energy to bring these things back together and recharge your battery. That is the ATP cycle. So when you study this, my recommendation, you can study how you want. But for me, the best way to learn this is start with the big picture and then start adding the details. Like I already said, the first thing you need to memorize is the fact that your cells need ATP for energy. And then, once we get back, we're going to talk about how is it made. It's made by cellular respiration of these are the 3 stages. And that's it. Don't forget the last word for attendance is to ask me a question about the material covered in this lecture. And I'll be in my office for office hours. You guys have a great weekend. And if you're online, don't forget to you have to send me those words before 9 a.m. For the rest of you, do send me the words before 9 a.m. It's extra credit for your attendance score. And of course, again, if you're watching the video at a later date, you also need the screenshots. Once you've got this far in the video, and then take the screenshots. Yeah, pick it. Thank you.